So it's now my pleasure to invite our chairperson for this session, Christine Vanabaya, Director of R&D Alliance Management at Ferring Pharmaceuticals. Christine holds a PhD in biophysical technology from Lund University and has worked for more than 20 years in the pharmaceutical industry with drug development in various therapeutic areas and administration forms. In her current role, she directs several alliances in the field of the human microbiota or microbiome and is today connecting to us live from Switzerland. So welcome, Christine. The floor is yours.
Thank you, Sarah. And thank you for inviting me to the Microbiome Summit. It's an honor, really, for me. I feel very strong links to the Copenhagen area, being Swedish, as I've been working in Copenhagen. And we also have strong links to Copenhagen via clinical collaborators. So I really feel honored to participate. I will do a small presentation related to a bit what I'm doing in Ferring Pharmaceuticals that I work for. And as Sarah already said, you can send in questions in the chat, and we will take them in the end of the session. So this session is, as you know, about antimicrobial resistance and the microbiome. And I'm going to repeat a bit. This has already been said, but I said to myself, there may be people who are attending this summit who are not so much into the microbiome as the presenters are. So just a small recap that the microbiome is not only bacteria, that's what you mostly hear about. It's also about virus and also fungi. And in fact, there are many other also organisms that we don't even know about today. So all these are part of the microbiome. And how they act on our body, as you know, the bacteria produce also. They produce metabolites, which actually act as precursors for many signal substances in the body. They also produce antimicrobial peptides and toxins, as you know. They also can, in fact, interact by themselves just by physically by adhesion, and can also affect different receptors by those means. The yeast and the fungi are also important. They are cofactors in many inflammatory and metabolic disorders, and they also help to modulate the microbiota. Viruses and bacteriophages. So bacteriophages, we already heard about them. We will hear more in this session. And bacteriophages are virus-like organisms that play an important role in regulating the microbial balance. And back to the metabolites, which are really important for our health, and they help to shape the way our bodies function by interacting with both our hormonal system, the neural system, and also the immune system. So that is very short about the microbiome. I also like to say just a few words on the antimicrobial resistance and some expressions related to this session. I looked up the Food and Drug Administration site, and the explanation of an antimicrobial resistance is the ability of a microorganism, which doesn't necessarily need to be a bacteria. It can also be viruses and fungi and other parasites to actually resist the effect of a drug. And if you look in the United States, it's mentioned on the FDA's homepage that today we have at least 2.8 million antibiotic-resistant infections every year, and more than 35,000 people in the US die per year as a result of this. Another source of good information is the Center for Disease Control in the US, called the CDC, which also talk about antimicrobials, drugs that treat infections by killing or slowing the growth of germs causing infection. And I think we all know, at least we think about antibiotics when we speak about antimicrobial resistance. But we also have antifungals, which are, of course, then drugs that treat infections caused by fungi. And this is scary information. The CDC report, the Center for Drug Control, who give out reports with some years. This one is from 2019 and the last one from 2013. And in the report, they list the organisms that are classified as urgent threats. It means that we are basically there when we don't have any drugs that can kill these organisms. I'm sure you know about them, like Clostridium difficile, for example. There are also some other. It's not a nice reading, and it's available for free on the internet if you are interested in looking more into this. There is also the serious threats and concerning threats and watch list. All with, I would say, the eye on those to be able to try to avoid that we get into a situation where we can't anymore kill those bacteria. If you look at the European Medical Agency, the EMEA, they don't say so much about antimicrobial resistance. Can be that I did not look in the right place. But what I found is interesting, and it's a draft guideline which actually is describing how to carry out studies to demonstrate the benefits and risks of antibiotics, and also get guidance on study design, etc. It means that they also start to seriously work on this. I don't say that they didn't do that before. Of course, everybody have a high focus on this antimicrobial resistance. Having said that, a few words about Ferring Pharmaceuticals and the microbiome. Personally, I work as Alliance Director in the microbiome field, which means that I'm directing a lot of development projects with a lot of external parties. This picture is showing a bit what we call our own microbiome ecosystem. And it all started back in 2008. If you go down to the left down corner, it's from collaborations with mostly a lot of French groups, academic groups. We worked with Institut Pasteur, University of Lille, and other academic groups in France. We expanded our collaboration with Intralytics, and we are now in a phase where we do a clinical trial together at the Mount Sinai Hospital in the US. And it's about an IBD-related bacteria that we try to target with Bacteriophages. In 2016, if you go up in the corner, the upper left corner, you see the logo of Karolinska Institutet. So we created a lab at the Karolinska Institutet. We call it the Center for Translational Microbiome Research, where we do basic research on the microbiome in both gastroenterology and reproductive medicine, maternal health. 2018, Ferring acquired Biotics, which is a US-based company doing fecal transplant for the indication of Clostridium difficile. And with the Biotics on board now, we say that we have our own microbiome innovation engine. And it's a very interesting times in Ferring because this field is really taking place now inside the company too. And the commitment with Karolinska is really a very basic science. We explore the microbiome and try to clarify what is normal and maybe not normal. If something is normal, which we almost, I think, already know that there is not, it's really up to the person himself or herself. And of course, we would like in the end to develop new therapies in these areas. This is a short slide on the IBD study, which we do with Intralytics and all these French collaborators. We are targeting a special kind of bacteria called IEX, stands for adhesive invasive Escherichia coli. And so we are in the clinical phase with this one. We also work with Intralytics on other indications, which are absolutely related to bacteria that we can't really anymore treat with antibiotics. We are very close to filing an IND for this project. We also have a third collaboration for Bacteriophages, which we do together with Intralytics and also the Eliava Foundation in Tbilisi in Georgia, former Soviet Union, Georgia. And it's about use of bacteriophages in women's health. And that's about Ferring. And now I would really like to introduce you to the speakers of this session, which is Lars Hjelback, Paul Ross, and Alexander Sulaklevice. And we start with Lars Hjelback, who is, welcome Lars. Lars is the professor at the Department of Biotechnology and Biomedicine at the Technical University of Denmark. He heads the Infection Microbiology Research Group. The aim of Lars' research is to better predict antibiotic-resistant evolution in Pseudomonas Aeruginosa and identification of alternative therapies against multi-drug resistant bacteria. The title of Lars' presentation is Identification of Anti-Virulence Compounds against methicillin-resistant Staphylococcus aureus from host-adapted bacterial pathogens. The stage is yours, Lars.
Thank you very much, Christine. Thank you for the nice introduction, and thank you for the invitation to be here at this great event today. So I apologize for the very long title here on my presentation. The key message is really anti-virulence compounds and bacterial pathogens. So what I will talk about here is our efforts to find or discover new types of microbial interactions or bacterial metabolites that can work as potentially work as anti-virulence compounds to limit the colonization and virulence of bacterial pathogens. And the take-home message from my presentation here is that the human microbiome is a reservoir that we haven't really fully explored for bacterial-produced chemistries that have bioactivities. For example, anti-virulence activities, but could also be antimicrobial activities. Right. So I'll start here with a little bit on bacterial pathogen and pathogen evolution. So bacterial pathogens, when they enter or colonizes the host, they are subjected to a range of different selective pressures from the host environment, from the antibiotics, and also from other microbes. So all these selective pressures means that the pathogen here, shown on the simple cartoon on the left, over the time of the infection will evolve, and you will generate a genetic diversity in the pathogen population over time. And this is particularly relevant in chronic or long-term infections, where this genetic diversity evolves. And we are interested in trying to explore this evolving diversity in the pathogen population, because it also may mean that these pathogens are then interacting in different ways with the other members of the microbiome. And by exploring these differences in interactions, we think we can find usable interactions, for example, anti-virulence compounds. Right, so the bacteria that I will talk about here is Pseudomonas aeruginosa. It's a devastating pathogen. It was on Christine's list, as she showed in one of her slides. Devastating hospital pathogen and associated with multi-drug resistance. But actually here in this talk here, this Pseudomonas aeruginosa is actually the good guy. It has been known for decades that Pseudomonas aeruginosa can produce a range of different virulence factors, and that many of these virulence factors have anti-staphylococcal activities. So they can kill Staphylococcus or inhibit growth of Staphylococcus. And you can see that by this very simple experiment shown here on the left corner. We played out Staphylococcus on an agar plate, and you apply a culture of Pseudomonas aeruginosa or supernatant of Pseudomonas, and you will see a nice clearing zone because of the killing of Staph aureus. And we also know quite a lot about the molecules that are doing this. This is shown on the right part of the slide. Different types of respiratory inhibitors, like pyocyanin, hydrogen cyanide and other things, and also enzymes and proteins like proteases and toxins, all together with the anti-staphylococcal activities. So when you begin to look at Pseudomonas aeruginosa and this genetic diversity that evolves in chronic infections, you often see this anti-staphylococcal phenotype has been lost. So most often, the genetic adaptation to the host by these pathogens are associated with a reduction or abolished production of these virulence factors. And if you take one of these host-adapted isolates from these chronic infections and do the same type of experiments as we did before, you no longer see this inhibition zone. So these host-adapted Pseudomonas isolates are now capable of more or less peacefully living together with Staphylococcus aureus. And the idea that we are pursuing here is really, now that we can have them cultivated together, and we have, or evolution has removed this normal antagonistic chemistry that is normally associated with Pseudomonas aeruginosa. Because of the host adaptation, we can now begin to screen for some of the underlying interactions between Pseudomonas and Staphylococcus aureus. And the way that we're doing it is we have selected a few virulence genes from Staphylococcus aureus, made a small platform of a selected few reporter genes. Some of the genes that we've selected are shown on the figure. I don't know if you can read that, but these are virulence genes that are part of the normal major regulatory network that controls much of the virulence in Staphylococcus aureus, and that is the AGR quorum sensing network. And I've highlighted some of the genes that we're using here in green and red. Right, so we have this small panel or platform of different reporter fusions, and some fusions are being highly expressed and some are not being expressed under different conditions. Here we're simply using an agar plate, and what can be done here is simply to screen our library or collection of different host-adapted Pseudomonas aeruginosa strains for variants that now changes the expression pattern of these reporter fusions. So, for example, on the right side, you can see one of these Pseudomonas isolates clearly producing a compound molecule here that interferes and reduces expression of the two top reporter fusions, but increases expression of the two lower ones. And particularly the top one is interesting because that's the RNA-free gene, which is a major effector molecule of this AGR quorum sensing system in Staphylococcus aureus. Right, so we can also begin to explore the diversity of this. So here I will focus on a particular clonal lineage of Pseudomonas aeruginosa, which is called DK2. We know that this can be found in a range of different chronically infected patients, and we can use our collection here to see what fraction of these chronic infecting isolates can now interact with Staphylococcus in a different way. And a substantial number, more than or about a third of these host-adapted isolates can then have a different response or elicit different responses on gene expression in Staphylococcus aureus. And we can also pass this down to the individual patients. So here on the right side, you see co-infecting sublineages of this particular DK2 isolate, and you can see that there are coexisting Pseudomonas strains, some that are highly inhibitory to virulence expression in staph, and some that do not have any effect. And I'll come back to that in a while. Right, so one thing is, of course, to have this screening platform with the other. We also, of course, want to have a deeper look into what is actually going on when Staphylococcus is meeting these host-adapted strains. So here we have done a simple RNA sequencing experiment done on this particular DK2 isolate grown on agar surfaces next to our Staphylococcus strain. And we can see multiple genes are being affected, not just the 4 that we are using in our screening platform. And when we then look more into that, we can see that many of the genes that are downregulated, these are actually virulence genes in Staphylococcus. So hemolysins, proteases, phenol-soluble modulins, for example, are being downregulated in production as a function of this particular interaction. And these genes here are all under the control of this chrome sensing system in Staphylococcus aureus, perhaps suggesting here that the molecule now being detected from this host-adapted Pseudomonas aeruginosa strain is targeting something very high up in the hierarchy of this regulatory network, preventing normal gene expression in Staph aureus. And we can also measure that by doing virulence studies. Here, data from a simplified virulence model, C. elegans. And if you look at the black curve, this is where Staph aureus alone is killing the worms quite efficiently with a LT50 of 2 days, as far as I can read here. But then the red curve is where you mix in both the Staphylococcus, the virulent Staphylococcus, and this particular DK2 isolate. And you see a much better survival of the worms in this case. So virulence, not just virulence expression, but virulence itself is also reduced in this type of interaction. And these data here is just to show that it's not because Staphylococcus aureus is being eradicated by Pseudomonas. We can still find Staphylococcus in this virulence model even after prolonged time. So it's not because of killing, it's because of an altered pattern of virulence gene expression induced by these host-adapted Pseudomonas strains. Right, so we can also go ahead and identify the molecules. So as I mentioned before, we had examples from this particular lineage where we had both isolates that was highly antagonistic for staph virulence expression, and we had isolates that had no effect on virulence expression. So just by comparing their LCMS spectra, we can see clearly some chemical differences in the production comparing these two strains. And also because we have these reporter fusions, we can go through a classical bioassay-guided fractionation procedure coupled with mass spec analysis to identify the actual compounds that are doing this effect on virulence expression in Staph aureus. Right, and because Pseudomonas aeruginosa itself is a bacterial pathogen, well-known pathogen, we also know quite a lot about the genes and their function. So it's not so difficult to go from sort of a chemical molecule and have an idea about the biosynthetic genes underlying this chemistry. So in this case, we could rapidly identify the biosynthetic genes and knock those out and show that not only is the production of this chemistry now abolished, but also in phenotype assays shown in the bottom right corner, with the mutant compared to the wild type, we can see that the mutant has lost its ability to interfere with the Staphylococcus aureus virulence gene expression. Right, so what we're approaching here, or what we're trying to approach here, is really to find molecules that can interfere with the normal virulence expression. And what we've shown here in this particular example is the identification of a small molecule produced by another pathogen, Pseudomonas aeruginosa. And the effect here is not just on one single virulence gene in Staph aureus. There is a broad effect on the whole panel of virulence genes. Most likely because this molecule from Pseudomonas is interfering with the chrome sensing system in Staph aureus. And these molecules that can sort of broadly affect the virulence gene expression may offer a new type of potential for targeting multi-drug bacteria, for example, MRSA in the case of Staph aureus. And several of the experiments that we did here was actually done on an MRSA-derived strain. So an anti-virulence strategy might be a tool that can maybe supplement the use of other antimicrobial drugs. Right, and I also want to highlight here towards the end that again, as I said in the beginning, that if we begin to look into the human microbiome and maybe not just the commensals, but certainly also the pathogens that have evolved all these different types of chemistries, not just to interact with the human host, but certainly also to interact with the other microbes inside the host. We might be able to find new types of interactions and metabolites that are useful. And this space, I think, is very poorly explored yet. I will also highlight here the fact that in this case here we took the advantage of taking a look into the genetic diversity of the pathogen, not just looking at a single sort of wild type isolate, but looking into this diversity of genetic variants that was evolving, and that was actually the key to identify this type of molecule. It would have been challenging to find it in sort of a standard Pseudomonas strain. And finally here, an acknowledgement slide. Several people was involved in this, and funding agencies as well. And with that, I think I will say thank you.
Thank you, Lars. Thank you for a very interesting presentation. It will be thrilling to follow your research. It's a very important field. Thank you so much. And with that, we will go on to the next speaker. Again, a reminder to send in your questions in the chat, and we take all the questions in the end of the session. So next speaker is Paul Ross, and he will be connecting from Ireland. Paul is the director of the APC Microbiome Institute at the University College of Cork in Ireland. His main areas of research is antimicrobials and anti-infectives, gut microbes and gut health, bacteriophage, and human and animal pathogens. The title for Paul's presentation is Mining Microbes for Antimicrobial Pharmabiotics, Bacteriocins, Phage, and their lysins. Welcome, Paul.
Hello, everyone. Just want to say how delighted I am to be talking to you today in Copenhagen, and I very much look forward to giving this talk. I'm from Southern Ireland, and I put up there, bottom right, a high-resolution map of where I'm from. I'm from Cork in the south of Ireland, from APC Microbiome Ireland, which is a large microbiome institute formed 17 years ago, composed of about 300 people. I'm talking today about mining the microbiome for antimicrobial pharmabiotics. And my partner in crime, Colin Hill, is my main collaborator. Together we lead a program called Microbes to Molecules within the APC. To start with, I just want to say something about the looming antimicrobial resistance crisis. If COVID-19 has taught us anything, it's taught us how vulnerable mankind is to infectious agents. Antimicrobial resistance is also one of the top global health threats that we face. Without urgent action, this could cause 10 million deaths annually by 2050. And to put that in perspective, about 2.8 million people have died from COVID-19 so far. And the world is running out of antibiotics, according to the World Health Organization. And what's even more concerning is the clinical pipeline that exists for them in a lot of cases are modifications of existing antibiotics. We believe that a lot of the solutions may lie in the microbiome itself. Our hypothesis is that we can mine the microbiota for what we call pharmabiotics, which are bioactives from mining host microbe dietary interactions as a tool to control pathogens and combat AMR. Within the talk, I'm going to give 3 examples of different types of pharmabiotics that we work on. Antimicrobial peptides from bacteria called Bacteriocins. I'm going to give 1 example of a live biotherapeutic that's in phase 1 clinical trials in the States at the moment. I'm also just briefly going to say something about bacteriophage and license, which we work quite a lot on. I did want to put this antimicrobial quest in context and say something about bacterial warfare that goes on within microbiomes. So the microbiome is a theatre of activity composed of trillions of microorganisms that are all in competition with each other. They're competing for nutrients, cross-feeding. They also produce a wide range of antimicrobial metabolites, including small antimicrobial peptides called bacteriocins, the subject of what I'm going to talk about. We work quite a lot on the rumen microbiome as well, where we estimate about 38%, nearly half of the bacteria, have the potential to produce bacteriocins. We've also shown in the past that bacteriocin production can be viewed as a probiotic trait. And in this, what you see here is just an image of live mice, and where the middle one has been treated with a bacteriocin producing culture that protects it against a Lux-tagged infection with Listeria. I see on the right-hand side, if you just delete the bacteriocin out of that strain, the mice are not protected. What are bacteriocins? Well, they're small peptides that are produced by some bacteria that kill others. They're ribosomally synthesized, which means you can engineer them, genetically engineer them, that is. They have high activity. A lot of them are post-translationally modified. You've basically two types, one that are heavily modified and one that are not. The modified ones are called antibiotics and sactibiotics. One thing about them is you don't get much resistance to them, and also they can be broad or narrow spectrum. And these are the types of applications of the bacteriocins that we work on. And we work on using them for skin protection, using them for modulating the human microbiome, and basically or to edit it or sculpt it. We believe they have a lot of potential. We have a live biotherapeutic for mastitis. We use them for methane reduction, animal feed, and also food protection. The prototypical best-known bacteriocin is called niacin, and you see a picture of it there. It's a 5-ring structure, quite heavily post-translationally modified. As I said, niacin itself is ribosomally synthesized, so there's a gene that encodes it and a number of genes that code for these unusual modifications. And we have basically self-cloned varieties of it that are more anti-listerial, more anti-staphylococcal, are better able to diffuse, better able to kill gram-negative bacteria. And so these bacteriocins can be self-cloned and genetically manipulated. And indeed, we have got approval for use of these in food systems because they're self-cloned, and also because you do find natural variants of niacin out there in nature. I just wanted to give another example of a recent bacteriocin we found called actifensin. This one was isolated on the cover of JBACT last year. We got it from basically sheep feces. And it's a very unusual bacterium. It's produced by Actinomycetes ruminicola, and it has a highly redundant structure. And like niacin, it kills a whole range of nasty and pathogenic bacteria, mainly gram-positive bacteria. And that highly redundant structure means this could be the basis for engineering new bacteriocins based on this structure and modification machinery. In this way, our group has built up a bacteriocin arsenal and what you see in this table here, we see a list of different bacteriocins down the y-axis and the kind of nasty bacteria that they kill, represented by a yellow square. See straight away niacin A and actifensin A killing all gram-positives that we tested against, where something like turasin down below is very narrow. And so these bacteriocins can be narrow or broad spectrum. And I give just two examples of an animal microbiome type applications for them. One is where we have a bacteriocin producing probiotic that protects animals against mastitis. And on the right-hand side, we have a collaboration called MetLab with New Zealand, Breaking Bad reference, whereby we're using a methane, we're looking at methane reduction using lactic acid bacteria, a project that's headed up by Catherine Stanton. I did want to give one example of how these bacteriocins can be used. And in this particular case, I want to say something about the target. In this particular case, it was Clostridioides difficile associated diarrhea. This is an antibiotic-resistant superbug, a spore former. It causes problems with antibiotics, with use of antibiotics such as vancomycin. And just put one statistic in there that's fairly staggering. 1 in 9 over 65s will die following 30 days of diagnosis. And this is one of the main targets for fecal microbiota transfers. We wanted to find a bug that would kill this organism. And what we did was we looked at other spore-forming bacteria. This is spore-forming Clostridia. So we looked at other spore-forming bacteria by simply looking at stool samples and ethanol treating them and see if we could find one that would kill a C. diff overlay. And what you see here, very much like an antibiotic, you see a huge zone that's produced by this Bacillus thuringiensis culture. In this case, a picture tells the whole story in that this is the original fecal sample that it was isolated from. And what you find is that basically in the zone, you're finding other isolates that it doesn't kill because it's very, very narrow spectrum. This is what it looks like. It's a new kind of class of 2-component, what we call SAC biotics. These have the unusual ring structures again in them with sulfur to alpha carbon linkages. What we do in the lab is we grow, we do quite a lot of fecal fermentations looking at new antimicrobials. In this particular case, these are human fecal fermentations. And what you see in the arrows here is where we inoculate them with C. diff. And you see, over time, they'll grow. And we in this particular case, we were taking purified churicin and comparing it to 2 antibiotics, metronidazole and vancomycin. You see, in all cases, they kill the C. diff. However, what you see is that this is the microbiota at a very high level, at a phylum level. And what you find is that churicin doesn't really alter the microbiota, doesn't cause the collateral damage that you see with the other antibiotics, metronidazole and vancomycin. Moreover, if you inoculate mice rectally with C. diff and then add churicin 60 minutes later, you see a huge reduction in C. diff. And so we have shown that it works in vivo as well. Within our microbiome center, we have formed a number of spin-outs over the years. One of them is a spin-out called, and Colin and myself are founder members along with Ronnie Farquhar, called Artigen Therapeutics, which is basically a company looking for microbiome-based solutions to infection. And in this particular case, we have a culture, a live biotherapeutic, that's in phase 1 trials in the US at the moment called ART24, that went through quite a large mining the microbiota and characterization phase, like I've outlined for churicin before that. Also, I want to say something about just a couple of slides where we're looking at basically bacteriophage and their license. Bacteriophage are bacterial viruses, and within these bacterial viruses, you have enzymes called lysins that break open the cell wall. We did some work some time ago, and this is basically looking at lung microbiota. In this particular case, this is a model for cystic fibrosis. In cystic fibrosis, often people get infected by Pseudomonas aeruginosa, particularly as in this case, where we're deliberately infecting mice. These are mucoid Pseudomonas aeruginosa that are tagged with luciferase. And so these are live mice again. In this particular case, they're nasally inoculated, and you see that the Pseudomonas is going down and infecting them in the lungs. If 5.5 hours later, you treat them with a cocktail of 2 bacterial viruses, bacteriophage, you see that the Pseudomonas is eliminated. And we believe that phage can be kind of targeted sharpshooters that can go in and eliminate infections like this in a very, very precise way. Also, there are 5 times more bacteriophages in your gut, as has been estimated, than bacteria in your gut. And we believe that the bacteria in your gut have a huge influence on your microbiome. Something that's very hot in the microbiome, as you know, at the moment, is fecal microbiota transplants. And we wondered, was there a role for phage in this? In this particular case, we had 3 donors and 13 successful FMT recipients over a year's period. The phage were transplanted during FMT. And phage can be highly donor specific, but what we found was basically that 1 year later in those recipients, these phage remained engrafted. And so the virome, if you like, was changed by DFMT. And this shows maybe a role for phage in modulating the structure of the microbiome. And so I just wanted in this talk again to say something about microbiome-based solutions that we call pharmabiotics. These are bacteriocins, live biotherapeutics, and phage, and they have the potential to shape the microbiome in ways maybe that antibiotics cannot. A lot of them, as I've shown, that can be narrow, like Turasin, avoiding collateral damage, or could be broad, like actefensin and Niacin. Phage can target undesirable bacteria in microbiomes due to their narrow specificity, and I gave an example of that, of Pseudomonas in the lung. And this leads, I suppose, to what we call precision biotics, where we can modulate the microbiome to influence how we respond to diet, our drugs, and fight disease. So lastly, I again would like to thank all my collaborators and friends within APC Microbiome Ireland, in particular Colin Hill and Catherine Stanton and Susan Mills. So thank you very much.
Thank you, Paul. Thank you for a very interesting talk. It's a fascinating field, and I really wish you all the best with the future for your research. It's time now for the last speaker in this session, and it's Alexander Sulakevitz, who will join us from Baltimore in the US. Alexander is the president and the CEO of a company called Intralytics. He's also an expert in phage technology, and he has been instrumental in, I would say, paving the way for the bacteriophages in the US via discussions and various approvals, also including INDs with the FDA. The title for Alexander's, or Sandro's, like we call him, is the title for his presentation is Bacteriophages for Modulating Human Microbiome, the Phage Biotics and Superbiotics, Approaches and Their Potential Applications. So very welcome, Sandro.
Good afternoon, everyone. I'm sorry I couldn't be there today in person to give this talk because of the coronavirus, but I still like to talk about good viruses, what we call bacteriophages, and how bacteriophages could be used to modulate human microbiome for various health benefits in a platform technology that we call phage biotics and superbiotics. And I explain this a little bit as we go along my talk. But let me start by maybe spending a minute or so about our company, the company called Intralytics. We're privately held, located in Columbia, Maryland. We are arguably one of the largest, if not the largest, phage manufacturer in the United States, possibly in the world, and I mean the FDA and USDA approved product. It's a high throughput robotic capabilities, and we have the largest manufacturing, as I said, both for liquid bacteriophages and dry powder bacteriophages. And this is all, and the facility is CGMP certified, meaning it meets the current good manufacturing standards. And all we do is work on bacteriophages. So just a quick refresher on bacteriophages. They are viruses, good viruses, again, that are estimated about 3.5 billion years old, arguably the oldest organism on this planet. They're also arguably the most ubiquitous organism on the planet. And I have a couple of slides later that will give you an idea of just how ubiquitous they are. They were discovered in 1917 by Felix Derel, who also came up with the name bacteriophage for bacteria eaters. And as I said, they're the most ubiquitous organisms in nature, and they play a key role in maintaining balanced bacterial population in every ecosystem, in a perfect predator-prey relationship, if you will. They're very effective in killing bacteria. They're also very specific in killing those bacteria. So bacteriophage that kills E. coli, for example, will not kill Listeria monocytogenes, and vice versa. And that specificity has some important implications in this whole platform technology, and I'll get into this a little bit as we go along. So for everyone working with antibiotics or other classical small molecule antimicrobials, bacteriophages are fairly large, but everything in this life is comparable. So if you compare them with many other organisms, including bacteria, the bacteriophages are small. And so this animation kind of gives you an idea how they compare in sizes with different organisms. And here you go, bacteriophage versus bacteria, about 70 to 200 nanometers in size. So they're very small in the microorganism world. I mentioned earlier they're the most ubiquitous organism on Earth. You can read the slide on your own. I'm not going to go into all the details, just to highlight a couple points. Human gastrointestinal tract contains about 10 to the 15 bacteriophages, and the total number of phages on this planet is about 10 to the 30, 10 to the 31st power. So that's really very, very significant number. So we literally live in a sea of bacteriophage. We also eat them daily. So 100% of unprocessed fresh foods contains billions of bacteriophages. And so we eat them and drink them every day. So getting back to their practical applications, remember I mentioned they were first discovered in 1917. Those are the years when antibiotics do not exist. And so because phages were so effective in killing bacteria, they were almost immediately used for clinical applications. So the first therapeutic use comes in 1919, just 2 years after their formal discovery. And the phages were used successfully to treat bacterial dysentery in 4 patients, 4 children actually, in France, and all 4 recovered well. Even though this was the first use of phages therapeutically, the study wasn't published until, I think it was 1926. And so the first publication that comes, that describes use of phages therapeutically is actually coming from Belgian group in 1921, when they used phages successfully to treat skin infections. And so several large companies, including Eli Lilly, manufactured phage products, and then they were heavily used in Soviet Union, Eastern Europe. Russian, German armies routinely used phage preparations during World War II. So it was a fairly common use until antibiotics became widely available, and then they kind of pushed out therapeutic phage use, at least in Western medicine. But the use continued in former Soviet Union, in Eastern Europe, and phages were used therapeutically in almost any imaginable route. Oral administration was arguably one of the most common, if not the most common, but they were also used rectally, locally, intravenously, and so on. So fairly common use clinically in 40s, in 30s, 40s in the United States, and continue to be used to this day in the former Soviet Union. And so the interest has been regaining lately because of the drug resistance, that people are beginning to take a second look at bacteriophages, and now can be used to treat drug-resistant bacteria. And so there are several clinical trials currently ongoing. And you can see we have, Intralytics has at least 2 clinical trials on this list, with 3 additional clinical trials in the pipeline to start within the next 24 months or so. And so, but what I'd like to talk more is a different trend of using bacteriophages when phages are used to fine-tune human or any mammalian microflora. And so This goes back to the title of this presentation when I mentioned phage biotics and superbiotics. And phages can be used to modulate microbiome on their own in approach that we call phage biotics, or they could be combined with existing or known prebiotics and or bacteria or yeast-based probiotics. And so that's what we call superbiotics approach. And that either of those could be developed as nutraceuticals, dietary supplements, or drugs. And they can have some very significant implications in human well-being. So I'd like to show you a couple slides now that show how phages can be successfully used to modulate microbiome, both using in vivo and in vitro model systems. So this is one of the earliest recent publications that describes use of Shigella phages compared to antibiotic ampicillin in treating Shigella infections in mice. And as you can see, the phages are numerically actually even more effective than ampicillin, but statistically about equally effective in reducing Shigella counts in mice. But there is an important difference in how they impact microbiome of those mice, and I'll show you this in one of the subsequent slides. But Shigella is not the only pathogen, and the treatment, of course, is effective if you administer phage a little bit before the challenge with Shigella or slightly after the challenge, indicating that prophylactic use is feasible, and also potentially therapeutic use is feasible, especially if it's shortly after infection, perhaps early onset of the disease. And this is not specific for Shigella. There are some other studies showing that you see similar results in other enteric pathogens. So this shows with Salmonella typhimurium. And this is a SHINE model, in vitro model, which has been populated with a microbiome of 3 different donors. And you can see that the phage administration reduces salmonella levels in all of those donor systems significantly. So it has a pretty good efficacy irrespective of the donor. So it's kind of a broad spectrum efficacy here in a sense. The phage cocktail, tentatively called FOP here, that contains salmonella phages is also shown to be fully protect and perhaps even reverse the salmonella-mediated disruption of intestinal epithelial cells, and also reduces the ability of salmonella to adhere and invade human intestinal epithelial cells. So the really important implications in preventing a salmonellosis in humans, perhaps in this case. So the important difference between other antimicrobials and phages is the impact on the normal microbiota. Because phages are so specific, they're expected to only kill their targeted bacteria and not impact anything else. And so this is the computer-modulated simulation that shows how what happens in a normal human microbiome if it is treated with antibiotics, and there are several antibiotics listed here, versus E. coli bacteriophages at high concentrations, shown here at 10 to the 9, or low concentration, shown here as 10 to the 4. And as you can see, there's a very minimal impact on microflora because of the bacteriophage treatment, and much more noticeable impact on microbiome because of the antibiotic treatment. Now this, of course, is a computer simulation, but we see essentially this very similar picture in various in vivo systems as well. So going back to the Shigella treatment, one of the earlier slides that I showed today, this now looks at the impact of the same Shigella phage cocktail at the film level on various bacterial species, bacteria in the human in the mice microbiome, and you can see that there is essentially no difference between the PBS control and the phage treatment. So this phage basically has undetectable impact on mice microbiome, and this is very similar in other mice treated with different phage cocktails and challenged with different bacteria. So this is a slide from another published paper, and you can see the publication citation down on the bottom of the slide, when 16S RNA sequencing was used to evaluate the impact of phage on the heat maps. And this slide shows this sorted by hierarchy. It includes treatment with FOP in dark green, control PBS, and then ampicillin in red. And you can see ampicillin has significantly more disturbing impact on microbiome compared to PBS and FASH. So PBS and FASH basically are equal. They basically don't have much impact on the microbiome at all. So same study, family heat map, just this time sorted by time. Again, ampicillin significant impact, FASH basically undetectable impact, showing that this is not only effective, and we showed some data in vitro and in vivo, and there's obviously a lot more data that I'm not presenting here today, yet it has very mild impact on microbiome. So which indicates that this technology can really be used to modulate the microbiome, fine-tune the gut or other microbiomes. And we could potentially develop series of products based on this platform that could be used to fine-tune microbiome for various health benefits. And so, and finally, the FASH cocktails could be also highly customizable. So you can design a FASH cocktail that kills specific bacterial species, bacterial subgroup of bacteria, subclonal groups, and so forth. So just to look at the slide, speaking about designing custom FASH cocktails. So this is something that is built by a program called Phage Selector that we've developed. And on the left side, you can see about 1,100 Salmonella strains tested against 26 bacteriophages. And you see that, if you combine all those 26 phages, we will kill about 95% of Salmonella strains. Now, if you start selecting the most potent bacteriophages, and on the right side of the slide, we now have the 6 bacteriophages, not 26, but only 6, against the same about 1,100 Salmonella strains. We still killed 93% of them. So it's very effective in killing. You don't need to have 26 phages to kill a large number of strains, and you have still significant redundancy, as shown in this color table, if you will, on the right side, that multiple strains are killed by more than one phage, which provides a good redundancy against perhaps resistance emerging against the bacteriophages. And if you start selecting specific serotypes between the Salmonella, say in this case Enteritis typhimurium, the same cocktail can kill 99% of those. So it's highly customizable. You can do a lot of things, and that technology allows you to do a lot of modulation of microbiome of humans. And when people talk about microbiome, the first thing comes to mind is the GI microbiome. And of course, phages, either phage biotics, phages alone, or superbiotics with phages with other probiotics could be used to target diarrheal diseases. Shigella is a good example of that. But they could also be used to manage bacterial vaginosis. Perhaps a good example here would be a superbiotics approach with Gardnerella vaginalis FASH combined with lactobacillus. As an example, could be other combinations, of course. Cosmetic applications, perhaps acne, atopic dermatitis, oral care against dental caries or gingivitis, and perhaps more importantly, non-infectious diseases, certain forms of cancer, obesity. So it's not meant to be all-inclusive, but what it shows that there are numerous opportunities. And as the microbiome field expands, there are lots of things that this technology can do, both the research-wise and product development-wise. So in conclusion, what I'd like to do is to thank you for your attention and ask you to think about your own research programs, your product development programs. And if you feel that phage biotics or superbiotics can be helpful, please get in touch with me. I would love to discuss various opportunities. So thank you again.
Thank you, Sandro. Thank you for this deep dive into the bacteriophages, and also for giving your own perspectives to the field. We are now going to go into a Q&A session, and we have got quite some questions. We will start with one which is directed to Lars. It's from Eric de la Fortelle. And the question is it possible to set up a high throughput screening to throw entire molecules at an assay to tame MRSA, virulence genes, or virulence genes from other bacteria? What do you say, Lars?
Yeah, so I illustrated here just the concept. Of course, this can be done in high throughput So if I understood the question was if this can be done with other bacteria also, yes. I mean, for these important pathogens, we do certainly know about which genes would be indicators for or can be used for the screening platform, and there's nothing to prevent that from being set up. And was the question also about chemical libraries? Because certainly, I mean, you can also screen through chemical libraries to see their effect, not just on killing, but certainly also on effects on gene expression.
Yeah. Thank you, Lars. We have another question, which is for Paul. It's from Carlos Leon Sobrino. What is the advantage offered by bacteriocins compared to antibiotics, which are also of microbial origin? I assume resistance could also emerge over time. And aren't they very sensitive to proteases? Paul, what is your answer?
Okay, 3 questions, very good ones though. I suppose the advantages is spectrum. We have a whole range of bacteriocins, and a lot of them are very, very different spectra to each other. And so a lot of the antibiotics are broad spectrum and cause collateral damage. So a lot of bacteriocins can be quite narrow because they've evolved basically to kill related bacteria that are in similar, nutritional niches and stuff like that. They also, the question is correct. You do get resistance to them, to some more than others, but the resistance for some of the antibiotics anyway can be quite rare, and in general, you don't get total resistance to them. As strains become more tolerant to them. In fact, even the strains that produce the bacteriocins are sensitive to them a lot of the time themselves, but have an immunity system to kind of overcome it a bit so they can grow. The last question, are they sensitive to proteases? Yes, because they're peptides, they're sensitive to proteases. But as I said, a lot of them are post-translationally modified. Those post-translational modifications can protect them quite a lot from proteases. And we have just done some, and these would be large animal feeding studies in pigs, to show that niacin, can be delivered to the intestine where it can modulate the microbiota. And that was something that goes against the dogma of that bacteriocins are just broken down in the intestine. For some of them, they're not.
Okay, thank you, Paul. There is another similar question, I would say, to Sandro. It's an anonymous question. What are the chances that the pathogens could evolve to become resistant to phages?
It's almost guaranteed. So chances are very high that they will eventually become resistant, and that will happen whether or not you use bacteriophages at all. It's just a part of the natural evolution. It's the biological arms race, if you will. It's been going on for billions of years. So it will eventually happen. There are some strategies to minimize the risk. Just to name few. Using of phage cocktails is one strategy. When you have 2 or more bacteriophages in a cocktail that kill the same strain. So if you have a mutation against one bacteriophage that makes it resistant against that bacteriophage, you may have another one that is still effective against the strain, so will still kill it. So a careful design of phage cocktail is one option. Perhaps as importantly, or maybe even more importantly, is the scientifically smart design of intervention strategies. So, for example, in a food safety business, and phages are used as part of the biocontrol, it's important to design the application that will allow phage treatment at the end of so-called epidemiological cycle. Just to explain it in a simple way, instead of going and spraying chicken house, for example, with bacteriophages, where we have very complex environment, many bacteria, rodents, and so on interacting, the chances of resistance in that setting are very high. So instead, it's preferred to spray bacteriophages at the end of the cycle. Let's say right before the packaging, when you spray phage on the food, it gets packaged and goes out, then the exposure is limited to that one package. So I use the food safety because it's much simpler to explain, but similar concepts should be designed for in vivo applications, including human therapeutic use. So wrap this up. Yes, it will happen at some point, but there are strategies to minimize the risk.
Okay, thank you. Another anonymous question, which I think is maybe for Lars or Paul. Are there PK/PD studies for bacteriocins?
Yeah, I can take that. This is pharmacokinetic and dynamic studies. Have they been done for bacteriocins? Not that I know of. They haven't. As I said, you know, a lot of the applications for bacteriocins have tended to go on because, you know, they can be broken down by proteases, have been topical. But as far as I know, those studies have not been done. Or I'm not aware of them anyway.
No. There is a question here, also an anonymous question. What is your opinion on synthetic phages, CRISPR? I can make a comment on that myself. I think it's very good technology, and evidently you can't maybe kill all organisms with phages, normal phages. I think there's one drawback though, which is important to think of, and that could be the regulatory way. It's difficult as it is to have live bacterial, live organisms approved by regulators. And my feeling is that if you go in to do genetically modifying, it will be even more difficult. I don't know if any of you guys have something to add on that. I'm sure you have.
I can add a quick comment, Christine. Over the years or decades rather, that we've been working with bacteriophages, we've been approached by several groups, academic industry groups, that have developed genetically engineered bacteriophages at the significant financial expense and time expense, and some very bright minds went into that work. And I'm yet to see a genetically modified phage that is superior to the wild type cocktails that we've been able to develop so far. So there are some reasons why people do it. Some of the reasons have to do with intellectual property protection. It's easier to patent, phage if you're genetically modified. It could be attractive from investment standpoint, raising funds. Finally, it's intellectually very challenging and rewarding. But in terms of the actual technical superiority, I am yet to see proof that the genetically modified phages are any better than, a carefully and smartly designed natural phage cocktails. So we'll see. It may happen at some point. I have not seen it so far.
Okay, it's time to wrap up. There are many questions left, which we didn't have the time to answer. I'm sure there will be other opportunities to discuss more about this interesting area. I would like to thank the presenters very much. Thanks to Lars, Paul, and Sandro. Very important field. I'd like to thank the audience also for listening and participating, and for all your questions. And also the organizers, of course. Very well done, very interesting meeting. Thank you so much.